Hello friends, I'm Shruti Parashar. Welcome to my channel Biology 360. Today I'm going to discuss the topic polymerase chain reaction, and that is PCR. This topic is important for students studying in ICSE, CBSE, IGCSE and HSC board, and for students preparing for NEET, AIMS and olympiad exams. A device used for gene cloning or gene multiplication in vitro. So in polymerase chain reaction technique, we make use of a device which is used for gene cloning or for multiplication of genes. In 1985, Carey Mullis made an important discovery in the form of an extremely powerful technique called polymerase chain reaction or PCR. PCR can generate a billion copies of the desired segment of DNA or RNA with high accuracy and specificity. So, by PCR technique, we can produce billion copies of DNA or RNA, and the technique is highly accurate and very specific. The process of PCR is completely automated. The device required for PCR is called thermal cycler. So the device or the machine that we used for PCR technique is called as thermal cycler. So in this slide, we see a device that we used in PCR. This device is called as thermal cycler. Requirements of PCR. DNA containing the desired segment to be amplified. We need several molecules of 4-deoxyribonucleoside triphosphates or DNTPs, such as we need DATPs, DCTP, DGTP and DTTP. Also excess of 2 primer molecules, heat stable DNA polymerase and appropriate quantity of magnesium ions. A DNA polymerase is a member of a family of enzymes that catalyze the synthesis of DNA molecules from nucleoside triphosphates, which are the molecular precursor of DNA. These enzymes are essential for DNA replication and usually work in groups to create two identical DNA duplexes from a single original DNA duplex. So this slide will tell us the different steps involved in PCR. Also the different PCR components. So we need DNA sample, primer, nucleotides, Taq polymerase enzyme, mix buffer and a PCR tube. And these are the different steps which are involved in PCR process. Here the first step is denaturing, wherein the double-stranded DNA is denatured at a high temperature of 95 degrees Celsius. The strands separate. The second step is annealing, which happens at temperature of 55 degrees Celsius. In annealing step, primers will bind the template. And the third step is the extension step. In this step, which happens at 72 degrees Celsius, there is synthesis of new DNA strand. Mechanism of PCR. At the start of PCR, the DNA segment and excess of 2 primer molecules, 4-deoxyribonucleoside triphosphates and the thermostable DNA polymerase are mixed together in Eppendorf tube, and the following operations are performed sequentially. The Eppendorf tube is the microcentrifuge tube. It's a very small tube in size which is used in labs that handle small volumes of liquid. Step 1. The reaction mixture is heated to a temperature of 90 to 98 degrees Celsius to separate two strands of DNA. This is called denaturation. Step 2. The mixture is allowed to cool at 40 to 60 degrees Celsius. That permits pairing of the primer to the complementary sequences in DNA. This step is called annealing. Step 3. The temperature of 70 to 75 degrees Celsius allows thermostable Taq DNA polymerase to use single-stranded DNA as template and add nucleotides. This is called primer extension. It takes around 2 minutes duration. One cycle takes around 3 to 4 minutes. To begin second cycle, DNA is again heated to convert double-stranded DNA into single strand. So the whole process in PCR, it approximately takes 3 to 4 minutes. In an automatic thermal cycler, the above three steps are automatically repeated 20 to 30 times. Friends, do subscribe to my channel Biology 360 and don't forget to click on the bell icon to receive the notifications on the biology videos. Also, please send me your comments and suggestions regarding the video as well. Thank you.